We want to keep moving right along, and we're going to now focus our energy here on batch-to-batch variability as it applies to allogeneic cell therapies. And so, Omkar, I'd like to talk to you a little bit about, just like we did for auto, what are the variability drivers for allogeneic cell therapies?
Great question. And I think we had a similar question from Nisha PR as well in the audience who asked a similar question. So theoretically, if you have the same donor, you are controlling the variability in the source material between batches. So technically, you would have significantly reduced drivers in your variability. But in practice, in reality, we have seen that's not always the case. So here's a few situations playing out, right? On paper, one allo donor is supposed to make enough batches to treat hundreds of doses. At least that's theoretically what the whole point of moving to allo is. But in reality, that number is significantly lower. In fact, I've heard of one of our industry partners saying that they get 4 doses from just one donor. So it's not really a multiple X amount of scale up, right? And you also need to maintain a pool of donors, not just one donor. You have, because once you've consumed cells from one donor, so 2 different things. Sorry, a pool of donors so that you have a wide range of potential recipients of the cells that do a one-to-one matching, right? Because you do some sort of compatibility HLA matching or some other means to see that the donor cells and the recipient's body are actually compatible and they accept it. So that's one. And secondly, once you have depleted your resources from that one donor, you have to go back and find that same donor. And that's not easy. That we have seen, like, people move, right? People go through various aspects, people go through life. At one point, that donor may have been healthy, and at the next point, when you're trying to source cells from the same donor, something else would have happened to that patient. So all of these factors actually do introduce variability even if you have a single donor-based cell therapy, which is allogeneic cell therapy. And I will also caveat this by saying one thing is that right now, because allo cell therapies are so focused on getting the product to the market, the go-to-market is their main target. They're going to be spending less time and money and efforts in optimizing and thinking about variability, and more time about how to get the product to the market. So that's one thing that we have seen that's slightly nuanced with allogeneic cell therapies versus autologous.
Yeah, sure, that makes perfect sense. Okay, Donna, I'd like to, again, sticking with allogeneic cell therapies, I'd like to get your take on what are the biggest challenges in batch-to-batch variation for allo cell therapy manufacturing, just kind of like we covered with auto. I'd like to have that same discussion with allogeneic cell therapies.
Obviously, it goes back to the same thing. It's that donor variability, but there's very consistent ways to address that. From the perspective and background that we have done with Triumvira, we spent a great deal of time understanding the donors for our targeted allogeneic therapy. That being said, this is another point where critical quality attributes come in play. We screened over 125 donors to then select down to 10, then to select 5 to build master PBMC cell banks. We now have attributes of incoming donors that we can build from for the future. And that is one of the biggest. Sustainability for an allogeneic program is hugely important. As Omkar mentioned, there is this rush to market, but there have been companies that have gone out there and it's like they're progressed forward and all of a sudden they have no availability of their donors for various reasons. Life happens, their donor pool dwindles away and they're acutely having to train that. So knowing what makes a successful allogeneic product is important. Then being able to work toward establishing a master PBM cell bank of those donors. Having gone through a pre-IND with the FDA, interesting enough, our proposal was to go with multiple donors and assuring that across those multiple donors, we got a consistent output in the product. That allowed us to screen out a lot of donors. Again, here's our expectations from mechanism of actions, expected functionality. So we developed those critical attributes in those donors and narrowed it down to 10, that we then developed multiple collections to show across collection of those donors, we get the same data set and product yield, and presented that to agency, and then have ended up establishing a rather significant pool of PBMCs that we can then start the manufacturing process. We do not pool across donors. We keep those donors separate, but we've shown that across those various donors and those various time of collections within a donor that we get comparable results. I guess I can say the good news for allogeneic is that, as Omkar mentioned, getting those large off-the-shelf batches is not realistic for cell and gene therapy, at least with the manufacturing mechanisms now. And for whatever your product is, most of these cells will not tolerate extensive long-term cultures without varying what your output is. For us, with a batch manufacturing, we get 20 to 25 doses, kind of depending on a few circumstances. But that being said, you can go to a little bit more of the traditional bioprocessing manufacturing, in that you can do batches and accumulate those batches into a master lot. So how you control that manufacturing is a little bit different than how you would obviously with an autologous product, but you can have an assembly line of devices that manufacture, so you have batch outcomes that you then combined and look at the expected results for a combined lot of product in the end. So that's a little bit more like traditional manufacturing processes. I think it's hugely important to understand, again, what the expectation of each batch is and have well-established expectations, so that you can then effectively combine those into a lot that you can have a significant number of vials to treat. And I say vials because we vial our product instead of bag. That you have a significant number to treat patients as you go through a study.